We have two talks today about non-human primates. One was to refine, and the next one is how can we replace non-human primates in science, especially now where we have this problem of not enough available non-human primates. Yeah, maybe that's a chance to move away from using them more quickly. Our next speaker who will tell us a little bit about this topic is Jarrod Bailey, PhD. He is the Science Director of Animal Free Research UK. He received his degree and his PhD both in genetics at the Newcastle University in England, and then spent 7 years investigating possible genetic causes of premature birth using human tissues and tissue culture. And for the past 18 years, Jarrod has evaluated the scientific validity and human relevance of animal experiments in many areas of biomedical research and drug and product testing. And he has promoted the use of human relevant methods in their place. He's a fellow of the Oxford Center for Animal Ethics. Jarrod, thanks for being here. The floor is yours.
Thank you very much, Kat. Thank you for a lovely welcome. I hope you can hear me okay, and I hope you can see my slides okay. Please let me know if there are any issues. So thank you, Kat. Thank you to everyone who's been involved for organising this conference, and I hope all the attendees will find it useful and interesting. I'm going to present a lot of information over the next 15 to 20 minutes, although there could be much more. I have cut this down. And a lot of the information I'm going to present, though by no means all, is the result of my own published work over the last almost 20 years. The volume of information is for one simple reason, really. I want to illustrate how much evidence is out there to support, I guess, the title of my talk, that there is a need for much more replacement of animal use and a need for much more future focus on replacement. And this is very simply due to the fact that data from animal experiments are poorly relevant and translatable to human biology and development. Now, I appreciate that there will be people here who disagree to some degree with that statement, but I believe that opinion is warranted by the available evidence and supported by an increasing number of scientists. So at the very least, I hope it should make for some good discussion afterwards, which I really look forward to. Kat gave you all an idea of my background. Most recently, about 2 years ago, I became the Science Director of Animal Free Research UK. Just to say that this is a charity that's been in existence for more than 50 years now under different names at some points, but it awards research grants to biomedical researchers at various stages of their animal-free research careers. And apart from that funding, we also engage in public affairs work, in educational programs, all to try to expedite the huge cultural shift in science that we believe is overdue and is warranted by available evidence. The next slide, just a second. Yeah, the next slide is really just from two repositories that hold the papers that have resulted from my work, and of course it's not any kind of feeble attempt to show off. It's an important response to what I've heard in talks before, which is the data I'm about to show you is in 60+ publications, book chapters, and so on and so forth. They are very widely accessed, downloaded, and cited. This is good quality, substantial evidence and the result of a lot of hard work. So I hope you'll bear that in mind, even if you disagree with it as we're going through. And we all know that there are two salient arguments in support of the 3 Rs, and particularly replacement itself. One is, of course, animal ethics and welfare, and the other is scientific. Are we really doing the best available science? Which means the most human relevant science, which also therefore leads to human ethics. Are we serving the best interests of humans in the best way we possibly can? Because if we aren't, we're actually causing human harm, both directly with the approval of dangerous drugs and dangerous chemicals, and indirectly via misleading data that do not lead to a better understanding of human disease and do not lead, therefore, to new effective drugs via that better understanding of human disease. And that also leads to drugs that are lost in development because they appear to have had unacceptable safety and toxicity profiles in animal tests. Now, before I get on to the science and the issues with human translation, I want to touch on briefly, which is a very, very important point that should always be uppermost in our minds, and that is animal welfare and the ethics of using animals, particularly, arguably, non-human primates, in perhaps the area that causes a significant, maybe the most amount of harm and suffering to non-human primates, and that is neuroscience research. So you may not want to look at the next slide, but I'm sure you've seen it all before. These images were taken from investigations a few years ago by my then employer, Cruelty Free International in London, which I think really shows what goes on in this type of research. We see monkeys with their skulls removed, so electrodes can be inserted into their brains. We see them in restraint chairs for other parts of the experiment, which are acknowledged as being extremely stressful. The monkeys are undergoing fluid and food restrictions so they can perform required tasks for the reward of a sip of juice or a snack. And these things are known to give some monkeys symptoms similar to human PTSD. Now, presumably, research like this has had the principles of the 3 Rs applied to it, arguably not very successfully. And I think we have to ask, can research of this nature ever successfully be refined to an acceptable degree? And can reducing the numbers of monkeys used in this way ever be thought of as doing enough? Now, this is obviously of huge ethical concern, probably the major reason why so many people oppose or have serious reservations about animal research, and especially monkey research. Now, a few years ago, I published a review of non-human primate research and non-human primate research in neuroscience, which concluded that based in the evidence of almost 200 referenced papers, there was a widespread exaggeration of the contribution of monkey research to human neuroscience, a widespread and significant exaggeration of the general human relevance of monkey research and monkey research in neuroscience, and again, widespread and significant exaggeration of the human benefits resulting from basic curiosity-driven science. At the same time as all of this, there was an underplaying of the contribution of human research to progress in neuroscience. There was an understating of the consequences of species differences between monkeys and humans and what that means to human translation. And there was also an underplaying of the degree of suffering for non-human primates used in this type of research. Now, all of this, of course, means that the harm-benefit balance that's fundamental to any approval or acceptability of, particularly of invasive research with monkeys that causes a huge amount of suffering. If the harms are greater than are widely acknowledged, if the benefits are less than are widely acknowledged, it tips the whole balance. And what this means is that there's a huge proportion of animal research, of monkey research, that would and arguably should never have seen the light of day. So I think the point is that far from applying the 3 Rs to research like this, it seems clear that some researchers, possibly even many researchers, are doing the opposite. They're seeking to justify their research where little or no justification can actually, in reality, be found. Now, in fact, a review commissioned by funders of research published back in 2011 in the UK agreed with much of what we said, even though that review was conducted and influenced and published by people with an interest in monkey research. They stated, in neuroscience research, in most cases, there was little direct evidence available of actual medical benefit in the form of changes in clinical practice or new treatments, and this might be because no link with medicine existed. In many cases, the panel of the view it would have been possible to carry out the work in humans. So much for the application of the 3 Rs. In vision research, which uses a lot of monkeys and is often particularly severe in terms of suffering, the actual and potential medical impact of most of the studies was low. And they were also compelled to recommend that further reviews of the outcomes, benefits, and impacts of monkey research should be carried out periodically, and that is not something that's been done. So our paper also made the case, and I'm going to whiz through these slides for time, that powerful techniques are available to keep neuroscience research, again, an area in which many monkeys are used and which often has severe consequences for the animals in a human perspective. Many techniques and approaches, such as scanning and imaging machines, ever improving in terms of their resolution, temporal and spatial, other non-invasive ways of tracking brain activity, all of these things are immensely productive and not affected by species differences, crucially, between humans and monkeys. And I'm going to get onto this in a few minutes. Now, notably, one of the claims made by monkey researchers in neuroscience is that you cannot get sufficient resolution using these techniques. So you must use monkeys to study down to the level of single nerve cells. Well, first, I know neuroscientists who disagree, who strongly believe, based on their own knowledge and experience, that the methods available for human research are sufficiently powerful, more than sufficiently powerful. And secondly, single neuron studies, if you really must do them, are done in humans. There are thousands of examples of single neuron studies in humans that have been done using research volunteers that have been undergoing brain surgery. Now, as I've said, we know we must keep a human focus because it's unreliable to apply monkey data to humans. And there are many and increasing species differences in gene complement and in gene expression between humans and monkeys. Many of these are known and acknowledged, which affect the function of neurons and indeed the brain as a whole. And there are a few examples here. Thousands of genes in every part of the brain that's been examined, for example here the cerebellum, are expressed differently, and this matters. At least 1/3 of many hundreds of microRNAs that affect gene expression diverge significantly between human and macaque brains, and dozens of these molecules are present in one species and absent in the other, and this matters. And something called AI, or adenosine to inosine editing, takes place at a much higher frequency in humans. Now, it doesn't really matter what that is. It's a post-transcriptional process in gene expression, but it takes place more in humans, especially in the brain, and it affects neuronal functions and neurological diseases by changing gene expression. Now, moving on to from neuroscience to animal testing of new drugs. Now, I know Thomas in particular talked an awful lot about toxicology. Most of my talk is going to be about research, but this is an important aspect of toxicology, drug development, that I did a lot of work on a few years ago. There was no evidence at the time we did the work I'm about to very briefly summarize, and I think still is no robust evidence to show that animal testing of new drugs has a scientific basis, that it's sufficiently predictive to actually defend. So we set off and did our own study using publicly available data and showed that, among other things, when a monkey test, we did it in many other species too, but when a monkey test suggested a new drug may be non-toxic or ostensibly safe, this adds almost no evidential weight to the existing probability, based on other tests and data, that a drug will also be non-toxic or safe in people. And we can quantify that. If you assume that a new drug has a 70% chance in humans of being non-toxic, if you get a negative test suggesting that non-toxicity in monkeys, that raises the probability of that drug being safe in humans from 70% to just 70.4%. That's negligible. It isn't worth doing that monkey test, either ethically in terms of animal welfare or in terms of the financial cost. And in fact, a couple of studies subsequent to ours resulted in a series of 4 papers, have shown the same thing. But animal testing of new drugs is still, I would argue, unscientifically defended. But fortunately, there's now increasing pressure on many sides and from many stakeholders to change this and to move away from these unreliable animal and non-human primate tests. Now, for the sake of time, and again, just to underline the volume of evidence available here, I'm going to whiz through a lot of other data. This is from a paper back in 2008 of mine. We've been looking for an AIDS vaccine for decades, and we still don't have one. Back in 2008, this analysis showed there'd been approaching 100 diverse, very different HIV/AIDS vaccines that showed promise, safety, and efficacy in animals, often in monkeys and chimpanzees. None of them worked in people. Now, I updated this in 2018, I think, in this recently published paper. Kat Herman is one of the co-authors. And there have now been around or more than 800 clinical trials of many more AIDS vaccines. We still don't have one that works in humans. There's an increasing opinion that Alzheimer's disease is floundering due to too much focus on animal models. The drug failure rate, the translational rate to human success, is approaching 100% for Alzheimer's drugs. Animals don't get Alzheimer's disease. And indeed, one of the mainstays of Alzheimer's research, the amyloid hypothesis, heavily based on animal research, is now being widely questioned with respect to its human relevance and calls to consider moving away from it. And we know why this is: species differences, the evidence is there manifesting in drug and vaccine failures, and indeed new news items reporting some new Alzheimer's drugs are framing the caveats about those drugs in terms of efficacy and safety, but also still questioning the amyloid hypothesis and if we need a different focus. Whizzing through, stroke is another area of failure. Many potential interventions tested on animals looking promising, and of those tested in humans, nothing significantly works. Parkinson's animal models are also being questioned. For those of you who might not know, the commonly used Parkinson's model is basically effectively poisoning monkeys' brains and believing that the symptoms those monkeys show are Parkinson's-like, but it isn't Parkinson's disease. And now there's an increasing opinion that the model is poor and not hugely relevant to the disease. And again, Kat and I were co-authors on this paper published last year with some really eminent Parkinson's researchers from around the world, saying that human-specific investigations in Parkinson's are not just possible, but they're necessary. We need to move away from the animal models. Some more examples of failure. Again, I'm going to whiz through this really quickly, this time from chimpanzees. They're our closest living relative. One might expect they'd be a great model for humans, of course, if we pushed ethics to one side. Now, I was involved with this project for several years that ran in the US, the only country in which significant amounts of invasive chimpanzee research took place. And this was a successful campaign. In spite of chimpanzee researchers saying that medical breakthroughs without chimp experiments would be impossible, the US Institute of Medicine agreed with us in our evidence back in 2011. The chimp research wasn't necessary and federal funding would be stopped. So first of all, there's the failure to develop the AIDS vaccine that I've already spoken about. Chimps were heavily involved in that and heavily bred for that purpose. As well as that, our program of research discovered that chimp research, claimed to be of vital importance by those who did chimp research, was often of so little importance it wasn't ever subsequently cited in scientific literature. And even when it was, it didn't contribute to any reported human medical breakthrough. What did contribute always was human-specific research. We saw that, contrary to claims, chimpanzees were almost never used in cancer research. Why? Well, because chimpanzees rarely get cancer, and even when they do, there are important differences with respect to humans with cancer. In hepatitis C research, chimpanzee use, if you look on the left-hand graph, had declined, while the use of human-specific methods on the right had skyrocketed. Many things that could be claimed could only be studied in chimpanzees, it turned out, with a bit of effort, could be studied using human-specific methods, and were being studied using human-specific methods. And we also reported why chimpanzee research wasn't reliably forming human hepatitis C. As with HIV/AIDS, where chimps could be infected with the virus, there were major differences in the pathology and the course of infection and disease. Something very, very different was going on in chimpanzees compared to humans. And we also looked at why chimpanzees were such a failure in research. It was claimed that they were almost genetically identical to humans. They weren't as close as it was often claimed by chimp researchers, and the same is true for monkeys. This might still seem impressive, but those small genetic differences mean a lot. These differences had been associated with many diseases and biological processes, including things like neurodegenerative diseases, immune system function, the study of infectious diseases, cancer, HIV, and so on and so forth. Further, even when humans and chimpanzees and monkeys share identical or almost identical genes, they often behave differently. They're expressed differently, which has huge consequences. Now, this slide shows that in many tissues, there is a large percentage of genes, hundreds if not thousands of genes that are expressed differently in many different organs of the body with major consequences. So I think that this evidence alone really underlines the call for more focus on replacement, a shift away from using animals as soon as we possibly can, very strongly, even if there were nothing else we could do. But there is so much we can do. And I'm going to quickly go through what these are. I'm sure other speakers will do that, so I'll go through these pretty quickly. We know that human cells and tissues of many kinds can be grown in the lab. We know if this is done properly and with care, and especially some of these advanced human cell and tissue cultures, three-dimensional cultures, organoids, organ-on-a-chip technologies, and so on, they are much, much more physiologically relevant than anything else that's gone before, something you can't really do with animal research. You can link, for example, these organ-on-a-chip technologies together for something that's called body-on-a-chip. These are already being widely used in research and drug development, and importantly, they can only get better, and they are getting better. Organoids, small, miniature, tiny little human organs are being grown using human cells, which function like physiological tiny human organs. They're being used across the world in many and varied areas of research instead of using animals, to mimic how new drugs are absorbed, metabolized, and excreted by human beings, and so on and so forth. And these can be seeded with cells from individual patients. So you don't just not have to worry about interspecies variability, translating across species barriers. You can factor in human variability into those techniques. Amazing networks exist and are being set up and improved constantly to facilitate the supply of human tissues to researchers. This has always been an excuse or a reason for people to stick with their animal models and to wait until they move into human-specific research. The supply of human tissues for these advanced cultures is increasing all the time. And we shouldn't overlook the power of computers, which can increasingly model well human organs and entire physiological systems. In some cases, as Thomas mentioned, they've been shown to predict the human toxicity of chemicals much better than animal tests. And such approaches are being used for many applications, including, for example, the repurposing of existing approved drugs for various new indications. So to start wrapping up, I think in summary, we have a problem. It's clear that there are many researchers, too many researchers, refining and reducing animal experiments where those refinement and reduction approaches are not warranted, but where replacement is. But where replacement is being overlooked and sidelined, arguably in favor of easier but much less impactful actions. The evidence I presented of the poor translation of animal research to human biology, and therefore ultimately to human benefit, and why that is the case, supports that alone. But there is more. For example, in the UK, and arguably elsewhere in the world, there are proposed programs of animal research must be licensed by the UK government. Now, parts of these license applications are published, including non-technical summaries of the proposed research, and these include a section on the application of the 3 Rs. Now, analyses of these non-technical summaries by my colleagues have shown that they often contain simple, unsubstantiated assertions that non-animal alternatives have been considered, but that there are simply none available. There's often no explanation of what or why, no detail, just simple, bland statements that do not withstand scrutiny. Now, of course, we must question what we do. We have to do that rigorously, deeply, and in detail. It's how science should be done. Many other areas of science and engineering test their hypotheses and test their models to destruction. Why not biomedical science? Why does this field seem to often and instead seek evidence to shore up hypotheses and models instead of asking questions of the validity of those hypotheses and models? For a long time, there existed hostility between those of us who seek replacement of animals in research and others who seek to justify and defend the use of animals. Now, that hostility still exists in places, but more than ever before, to finish on a positive note, I and many others believe there is common ground and a willingness to work together towards common goals. And this is where I'd like to finish my talk. There now exists a situation where we can help and are helping each other do what we want to do. We want to do good science, and we want to move away from animal use. I'd like to give some examples. Recently, a Dutch initiative called the Helpathon has been helping animal researchers, aided by other people, including biomedical researchers with broad and diverse expertise, including in new approach methodologies, to shift their research focus to those new approach methodologies, to find a way to address their research questions without using animals, all in a constructive, helpful, and non-judgmental way. This photograph on the slide shows the first UK Helpathon that we hosted just at the end of last year, which involved helping researchers in London develop and use 3D advanced tissue cultures to research inflammation in arthritis instead of using animals. The researchers loved the experience, and we have more planned. We have helped young researchers at our Animal Replacement Centre of Excellence, or ARC, in London to develop 3D human models of tumour microenvironments to study metastasis, how cancers spread. And we've worked with researchers who are repurposing existing drugs for new indications, such as SARS-CoV-2 brain infection and Parkinson's disease, using artificial intelligence linked with patient-derived organoid cultures. The way forward is clear, I believe. Replacement is possible if we try hard enough and if we help each other. But the first step has to be an acknowledgement that this needs to happen and that all efforts will be made to make it happen. And we're all winners when it does. Animals, people, patients, scientists, and science itself. I'd like to end with my usual slide, as this is my current and former furry and feathery friends who live with me up in the far north of England, in the wilds of the North Nines, who make me feel privileged and lucky to have them every day and remind me that animal welfare has to be central to everything we do. Thank you so much for your attention on what I think has been a bit of an onslaught of information. Just to direct you towards our website, which is on the slide. If you want to read any of my papers or know more about the research I've presented, it's on my ResearchGate profile there, or you can email me at that email address, and I'd be very happy to hear from you. Thank you so much.
Thank you very much, Jarrod. This was a wonderful overview, and thank you for also pointing people to all the information and papers. I will see if there are questions coming up. I would say let's have a 5-minute break now. I'm sorry, we are running a little bit behind. And then we continue with the next talk. And then we have this panel discussion with our speakers where also questions can be posed in the end. So if you have questions in mind, maybe just ask them then. It will be the panel about why some 3Rs are not adopted and others are. And yeah, I hope we have some time for discussion then.